All right, today we'll be talking about the renal tubules and its mechanisms. First of all, a recap on nephron anatomy, and that is that the nephron can be divided into 5 segments, each having their own purpose, and that are the proximal convoluted tubule, descending loop of Henle, our ascending loop of Henle, our distal convoluted tubule, our collecting tubule, and collecting ducts. So before we begin, there are 2 types of nephrons. We have our cortical nephrons and our juxtamedullary nephrons. Our cortical nephrons are named aptly so as they reside within the cortex of the kidney. And the main differential between this and our juxtamedullary nephrons is that our cortical nephrons have short loops of Henle, whilst our juxtamedullary nephrons have long loops of Henle. This allows for greater concentration of urine via greater osmolarity differentials that allow for more water to be reabsorbed from our luminal fluid. So our proximal convoluted tubule. It is responsible for the reabsorption of approximately 65% of our sodium and water, our chloride ions, our bicarbonate ions, our potassium ions, as well as other molecules present in the fluid. In terms of specializations, the membranes of the proximal convoluted tubule is covered in brush borders, which have protein carriers present on it. This allows for greater surface area, and as we know, greater surface area equals to greater rate of diffusion, allowing for fluid to be reabsorbed back into our body. In addition, this area would also have large numbers of mitochondria to facilitate the active transport that is required to reabsorb these compounds. Now looking at the next segment of the nephron, it is known as the loop of Henle, which can be divided into 2 segments, the thin descending loop of Henle and the thick ascending loop of Henle. The thin descending loop is only permeable to water, whilst the thick is not. It has thin epithelial cells and its purpose is to reabsorb water aided by interstitial salt content, causing an osmotic gradient. The thick ascending loop actively reabsorbs our sodium, potassium, and calcium ions, as well as bicarbonate. And it is due to this reabsorption of these ions, which creates the countercurrent system by producing a osmotic gradient that results in fluid or more water to be reabsorbed in our descending limb. Next, we'll explain the countercurrent multiplication system. The deeper you go into the interstitial medullary, the higher the osmolarity is. And as such, the greater concentration of urine becomes as water travels to areas of high osmolarity, and therefore we have greater reabsorption as we proceed further and deeper into the medullary. But how exactly does this work? So filtrate enters the descending loop of Henle at approximately 300 milliosmoles. At this point, the difference in osmolarity between the inside of the tubules and the surrounding medullary tissue is equivalent. As water is reabsorbed across the descending loop of Henle, the osmolarity of fluid within the descending limb increases. The high osmolarity fluid then enters the ascending limb, which is impermeable to water, and where sodium ions, our potassium ions, our calcium ions are actively pumped out of the fluid and into the interstitium. Now, there is a higher interstitial medullary osmolarity, causing there to be a greater osmotic gradient, resulting in more tubular fluid to be reabsorbed into the body in our descending loop of Henle. This process then repeats itself until a maximum osmolarity, interstitial osmolarity of about approximately 1,200 osmoles is achieved, resulting in our maximal concentration of urine. This value is dependent on the length of the loop of Henle, and that's why some animals, especially those found in the deserts, are able to survive on so little water, as their loops of Henle are much longer than that of humans, allowing them to concentrate urine further by extracting more water out of the tubular fluid and back into the body. This is also why the cortical nephrons are worse at concentrating urine in comparison to our juxtamedullary nephrons. Next, we have the distal convoluted tubule, where these specific ions are reabsorbed, as well as our water, in addition, key hormones that are able to modulate these functions are aldosterone and our parathyroid hormone. In terms of our aldosterone, it acts to trigger the reabsorption of sodium and secretion of potassium due to increasing the expression of our sodium potassium pumps. This results in increased osmotic gradient and therefore increased reabsorption of water. Parathyroid hormone increases transcriptional factors that increase the expression of calcium transporters on the surface of calcium transporters and therefore increases the reabsorption of calcium. And it's important to note this as this may lead to arrhythmias in the heart, which could be life-threatening. All right, next we have the collecting tubules and the collecting ducts, which have similar functions as the distal convoluted tubule, reabsorbing our sodium chloride and our water molecules, in addition to urea. Aquaporins are a specific protein channel found only in the collecting ducts and tubules of the nephron and allow for the reabsorption of water. The relevance of these protein channels is due to their direct modulation via our antidiuretic hormone or vasopressin. And an increase in ADH will result in an increase in expression of aquaporin and thus increase in water reabsorption. ADH is secreted by the body in a state of dehydration and is what you want and it's exactly what you want when you're dehydrated. You want more water to be reabsorbed and from our urine and less to be lost. Additionally, like before, aldosterone also plays an active role in this area. And the next slide is a summary of what has been discussed today and a very good summary diagram to remember and keep in your mind.